Homologous recombination. Homologous recombination constitutes the only way to fix double-strand breaks in our DNA. It also constitutes the way in which we generate variability in our species, as this is the way we mix our genetic information to generate our gametes. The type of injury that results in the simultaneous breakage of both strands of DNA is exemplified by exposure to ionizing radiation. When both strands of DNA are broken simultaneously, the cell can deal with the damage in two different ways: by using homologous recombination or by using non-homologous end joining. Homologous recombination requires the presence of a normal homologous DNA strand. This implies a strand of DNA sharing extensive sequence identity with the broken DNA and containing no breaks on its structure. Upon homologous recombination, the broken DNA is fixed without any losses in DNA information. Non-homologous end joining, on the other hand, does not require another strand of homologous DNA, but results in the loss of bases at the broken ends, thus introducing mutations into the DNA. The first event during homologous recombination is the breakage of the two strands of the DNA. This triggers a nucleus digestion of the broken strand. The broken strand then gets paired with its undamaged homologous double-stranded DNA and initiate the process of strand invasion, which generates two templates for DNA synthesis. The broken ends of the DNA are then used to prime DNA synthesis in a manner similar to what happens during DNA replication. Then, ligase closes the remaining nicks in the backbone of the DNA, producing two intact, break-free molecules of DNA. These crossed molecules of DNA form what is known as a holiday junction. Holiday junctions are resolved by the action of specific enzymes, and depending upon how they are resolved, they can generate either non-recombinant heteroduplexes or recombinant heteroduplexes. In mammalian cells, the process of DNA recombination is initiated by the binding of a protein known as RAD51 to single-stranded DNA. This produces a RAD51-coded single-stranded DNA molecule capable of binding to double-stranded DNA, therefore forming a non-base pair complex that triggers the invasion of the single-stranded DNA into the homologous double-stranded DNA molecule. This provides a target for the action of DNA polymerases and ligases, thus resulting in the formation of the holiday junction. The enzymatic machinery in charge of resolving holiday junctions in mammalian cells is not well characterized, and the identity of the proteins involved remains to be established.